Okay, thank you. So I'm a PhD student at the Bogolubov Institute for Theoretical Physics in Kyiv, Ukraine, but as I have been said, I currently at the Institute of Experimental Medicine in Budapest in Hungary. And today I would like to present our joint research with my PhD supervisor, Alexandra Vidibida. So we will be talking today about the impact of delayed feedback inhibition on neural activity. And why is this topic important? So as you may know, for brain, in order to function properly, there must be a balance between excitation and inhibition. However, a lot of recent experimental evidence indicates that cortical disinhibition is important for many cognitive processes like learning, memory, attention, social behavior, and so on. And cortical disinhibition is a transient ceasing of inhibition or activity of inhibitory neurons. And this cortical disinhibition can be achieved in different ways, for example, at the level of local circuits. And in one of the cases, how this can be achieved at the level of focus circuits. For example, one type of inhibitory neurons, that is somatostatin expressing interneurons, they are inhibited feed-forwardly by other type of inhibitory neurons, parvalbumin expressing interneurons. However, it is unclear how the inhibition of parvalbumin expressing interneurons is achieved. However, recent work by Deleuze and colleagues provides us with a possible answer to this question. And the previous experimental work showed that parvalbumin expressing interneurons in the cortical areas tend to send synapses not only to other neurons, but also onto themselves. And these self-synapses are called autopsies. So Deleuze and the colleagues showed that for parvalbumin interneurons, the main source of inhibition is autoptic self-inhibition. And all of this experimental evidence motivates us to study the impact of inhibitory autopsies, or in other words, delayed feedback inhibition on neural activity. And we approach this research as follows. So we consider some class of non-adaptive spiking neuronal models. And we do not specify the exact neural model. However, we specify the conditions of the set of neural models that we consider, and I will talk about this set of neural models in more detail a little bit later. So we consider that some neuron is stimulated with a random sequence of identical excitatory impulses, and this sequence is random, and under this stimulation, this neuron generates another sequence of spikes. And also, since we are interested in neurons with inhibitory autopsies, we assume that every time neuron generates a spike also enters the delayed feedback line or autops. And the impulse in the feedback line needs delta units time to reach the neural input and act somehow on this neuron. So as regards the input stimulation, so as I said, we consider that this is a stream of identical excitatory impulses, and we assume that this stream is a realization of some stochastic renewal point process. And we assume that we know the distribution of interspike intervals for this input stream, and we denote this distribution as P of t, where t is an interspike interval duration. And our goal is to derive the distribution of interspike interval for the output stream for this neuron with delayed feedback. We denote this distribution as P of t. However, we would like to express this distribution P of t in terms of the distribution for that same neuron under this same renewal stimulation, but without delayed feedback. And this distribution for such neuron we denote as P0 of t. So to sum up, we assume that we have distribution for input stream P of t, that we have distribution for a neuron without delayed feedback, P0 of t, and also that we know the delay of the feedback, delta, and we are looking for the distribution for a neuron with delayed feedback, P of t. So as I have promised, now a little bit about the set of neural models that we consider. So this set of neural models include such popular models as perfect integrator, leaky integrated fire neuron, and also binary neural model. And we assume that the class of neural models that we consider is, it satisfies these 5 conditions, which includes that neuron is deterministic, that it can be triggered only at the moment of receiving an input impulse, and also that just after triggering, neuron fires a spike and immediately it appears in its resting state and remains there until an input impulse is received. Also, we assume that we know the distribution for renewal input stream P of t, and also the distribution for output stream P0 of t. As regards the delayed feedback inhibition, we assume it has these 3 following properties. The first one is that the time delay in the feedback line delta is constant. The second one is that the feedback line is able to convey no more than 1 impulse at a time. And the third one is that after receiving an impulse from the feedback line, the neuron appears in its resting state and that impulse is immediately forgotten. So now let's move to how we approach the finding of a distribution of ASIs for a neuron with delayed feedback. But firstly, I want to emphasize 2 things. That the first one is that we study neural activity in stationary regime. And the second one is that under all the assumptions that we have made, always at the beginning of the interspike intervals, there is always an impulse in the feedback line. And let's say the impulse in the feedback line needs the beginning of the interspike interval. This impulse needs S units of time to reach the neuronal input. And we will call this variable S as time to live. So now we can find the distribution of ASI for neuron with delayed feedback using this expression. And here there is integration over different times to live. F of S is a probability density function of times to live as in the stationary regime. And the second distribution is a conditional probability density to get the output in ASI of duration t, given that at the beginning of the ASI, there was an impulse in the feedback line with time to live s. So now let's find the expression for these 2 distributions. So firstly, let's talk about the distribution of times to live f of s. So we have proved that if this condition on the distribution for a neuron without feedback, P0, and the delay of the feedback delta is met, then for any initial conditions, stochastic activity approaches a stationary regime characterized with such distribution of times to live f of s, which consists of regular part and singular part. And here you can see the expressions which will help to find these different parts of the distribution f of s in the stationary regime. So as regards the conditional probability, with this conditional probability, the expression for it you can see here. Here, chi is a Heaviside step function. Here is a distribution for a neuron without feedback. Also, here you see one of the two auxiliary distributions that we need to find our distribution. So this one is a distribution of output psi for a neuron with excitatory instantaneous feedback. And this distribution you can find solving these integral equations. And the second auxiliary distribution is this p tilde 0, and it allows to calculate the probability of obtaining an input impulse at time s prime that will not trigger the neuron without feedback. And this auxiliary distribution you can find solving this integral equation, which requires us to know the distribution of input ASIs and also output ASIs for a neuron without feedback. And one thing I want to note here is that if we consider as an input a Poisson point process instead of general renewal point process, this expression simplifies a lot. And you can see here how it looks in case of when its input process is Poissonian. And the reason why is that is because if you look at the intensity of point process, when the point process is homogeneous Poisson process, intensity is constant. However, it's not the case for the renewal point process, because intensity of renewal point process depends on how much time passed after the last event. So returning to the distribution of ASI for neuron with delayed feedback inhibition, this expression for this distribution P of t is different for different values of interspike intervals. So we have this expression when ISI duration is less than delta, and we have this expression when ISI is longer than delta. As we can see, the resulting distribution has jump type discontinuity at the point that corresponds to ASIs equal to delay at the feedback line. And to illustrate these general analytical formulas, I would like to show you the results that we have obtained for the model of body neuron with threshold 2 under Erlang 2 input stream stimulation. So here in the upper left corner, you see the plot of the distribution for input stream. So interspike intervals here are distributed according to Erlang 2 distribution, and you can see the plot here. Here on the right, you can see the distribution for the neuron without any feedback. And here on the left, you see the distribution for a neuron with delayed feedback inhibition. And in this case, we set the delay of the feedback at 2.5 milliseconds, so at this point, and you see there is a jump type discontinuity at the distribution. And this jump type discontinuity makes it impossible to model this distribution as any simple distribution. And also here you can see the plot of the results that we obtained by numerical simulation of stochastic neural activity to verify our analytical results. So to sum up, like two important points of my talk are that the first one, that you can find the distribution of ASIs for neuron with delayed feedback inhibition if you know the distribution for input renewal process, distribution for output ASIs for the neuron without any feedback, and also if you know the delay of the feedback, and the resulting distribution for a neuron with delay feedback will have a jump time discontinuity. So if you are interested, you can look at our preprint at Archive to find out more details of this work. So thank you very much. And it will be my pleasure to answer any of your questions.
Thank you, Ola. Are there questions or comments? We have time for answering.
There is a question from Cindy.
Okay, please, Cindy.
Sorry, can you repeat, please, one more, because my connection
is not very... I like your talk very much. Thank you, Oha. I like your talk very much. And I was wondering about the delay being just through one neuron. It occurred to me that it would be interesting to find out what happens if the delay is actually through a sequence of neurons as well.
Thank you very much for your question. So depending on which sequence of neurons you consider, for example, you can consider, so in our work here, we considered an inhibitory neuron with self-feedback. But instead, you can consider, let's say, excitatory neurons that is connected to some other inhibitory neurons that is connected to these original excitatory neurons. And I think under some conditions, you can use our results to describe the activity of these excitatory neurons that is connected with inhibitory neurons.
Thank you.
Other questions, comments? I can ask something. Have you tried to check on data your model?
Mm-hmm. Yes, thank you very much for your question. So we have not tried to check it on the experimental data because, so the thing is, if you look at the experimental data that that studied the of inhibitory neurons, you can see that this delay is very short. It's usually less than 2-3 milliseconds. And if we look at the resulting distribution, we see that the peculiarity that is
we don't hear anymore.
Is that for these short interspike intervals, you can have like other factors that will influence this distribution. So that's why we have not tried to apply this result to the experimental data yet. And also, there is not a lot of experimental data about autopsies yet.